Hi, today I'm going to talk about Mendelian genetics. Gregor Mendel, who lived during the year 1882 to 1884, is considered to be the father of Mendelian genetics. He was the one who is responsible for the laws governing inheritance of traits. Mendel was an Austrian monk. He was the one who studied the inheritance of traits in pea plants. He developed laws which described the patterns of inheritance which formed the basis of the genetics that we know now. Mendel's work was not recognized until the late 20th century. He worked mostly on pea plants and then tested them and found different hypotheses which formed the basis of the Mendelian genetics that we know now. So he is called the father of genetics. This picture shows the site where Gregor Mendel had conducted his experiments near his lab. So here I'm going to talk about a few things which are called as the particulate inheritance. Mendel stated that physical traits are inherited as particles. These particles are nothing but the alleles in the chromosomes present in the DNA. A few terminologies to remember before we go into the topic. The first one is trait. Trait is nothing but a character that can be passed on from one generation to the other or from one parent to the offspring. Hereditary is passing of traits from parent to an offspring. Genetics is the study of hereditary. As we go into studying genetics, we have to learn about genetic crosses. The first one is called monohybrid cross, a cross that involves a single trait. Example, the color of a flower. A dihybrid cross is one that involves two traits, two specific different traits, that is flower color and plant height. So Punnett Square is one which is used to solve genetically inherited problems. So it looks like this, where there's a checkerboard with two gametes placed on either side of the checkerboard. As you see here, to the left we have the male gametes that are written here and the female gametes on the right side. This can be interchanged anyway, but here we show it in this pattern. If we cross these two gametes, we get a particular character here which is put into one square. So how to make a Punnett square? So here we have to take two parents, one is the male and the female, and then make a grid and put the alleles alongside the square. So fill the grid by crossing each one of them as it is shown here. The first allele with the first allele of the father with the first allele of the mother, or the male with the female, and then you get a Punnett result. Now we go on to another terminology which is called as alleles, which are two forms of a gene. One can be dominant and the other can be recessive. Dominant is one which is stronger or which exhibits its character, which is represented by a capital letter. The recessive gene is one which is masked by the presence of the dominant gene, and it is represented by a lowercase letter. Genotype is the genetic combination of a trait. Example, RR, both capital RR, one capital and one small RR. The capital letter shows the dominant character and the small letter shows the recessive character. Phenotype is the physical appearance of a particular trait, which could be the color or the shape. So genotypes of alleles: R for red flower, R for yellow flower, the small r. All genes occur in pairs, so two alleles affect a characteristic Possible combinations are the genotypes red, both characters are in red, then red, that is the heterozygous condition where R is the dominant character and the yellow R is the recessive character. And then yellow, we have both the recessive characters which are shown in the lowercase letter. Now homozygous, gene combinations involving two dominant or two recessive genes. It could be either both the dominant RR or the recessive RR. In both these conditions, these are called as pure. Heterozygous, gene combinations of one dominant and one recessive allele. Here, as you can see in the example, here one capital R and one small r. Mendel worked with pea plant. Why did he choose pea, that is Pisum sativum? These can be grown in a very small area, produce lots of offsprings, and can be self-pollinated to produce the generations that you require, and can also be artificially pollinated. The pollen contains the sperms produced by the stamen. The ovary contains the eggs which are found inside the flower. These pollen carry the sperm and the eggs for fertilization. Self-fertilization can occur as well as cross-fertilization could occur. Mendel did these experiments with himself. He hand-pollinated using a paintbrush, or he would snip the stamens to prevent self-pollination. He traced these things through several generations. Mendel began his experiment. He produced pure strains by allowing them to self-pollinate. He used 8 pea plant characters or traits, as we see here: seed shape, seed color, pod shape, pod color, seed coat, flower position, plant height, and flower color. So this is a table that is taken from one of the literature that was available. So here you can see the seed shape, which is smooth and wrinkled. Some of the pods are smooth and some are wrinkled. And the seed color, yellow and green. The shape of the pod, smooth or inflated and wrinkled. The pod color, green or yellow. So based on these characters, Mendel started his experiment and did his experiment, which is shown here. So now the theoretical expected ratio of plants producing round or wrinkled seed is 3 round, 1 wrinkle. Mendel's observation was 2.96:1. So P1 is nothing but the first generation or the parental generation in a breeding experiment. F1 generation is the first generation of the offspring in a breeding experiment from breeding individuals from the P1 generation. F2 generation is the second generation. So here we see the crosses that can take place. First is the pure plant cross, the dominant with the recessive, which produces hybrids. Then you get the F2 generation with all the characters like 3 tall and 1 short. Now we'll look into the monohybrid cross. We'll take a single character, that is the seed shape, round and wrinkle. So if you put the Punnett square, we find here that the alleles that are formed, the genotype is all round and all alike. The phenotype is also all alike. The monohybrid cross can be summarized like this: the genotypic and the phenotypic ratio all looks similar. The monohybrid cross, the seed shape, if we take the heterozygous condition, both similar characters, the ratio is 1:2:1 or 3:1, round and wrinkle. The monohybrid cross review, the heterozygous is 50%, 25% homozygous dominant, 25% recessive. Mendel then went forward. He did a test cross, which we shall see in the next lecture.